guys, it's me Lydell. Welcome back to my channel. If you're new here, please subscribe and click the notification bell for you to be updated for my upcoming videos. Hi guys, I'm Lydell Molona, and now I am discussing about the microbial genetics. As we all know that microbial genetics is a subject area within microbiology and genetic engineering. It studies microorganisms for different purposes. The microorganisms that are observed are bacteria and archaea, as well as fungi and protozoa are also subject used to this field. The chromosome is one long double helical strand of DNA, and the DNA is tightly wound up and associated with many DNA-binding proteins, in which these proteins are essential for carrying out the two functions of the DNA: the replications and the expression, in which can be divided into two parts: the transcription and the translation. The DNA-binding proteins include the DNA polymerase, the RNA polymerase, the endonuclease, the ligase, the helicase, and the histons. When we say microbial genetics, it tackles about the genome, in which it is all the genetic that a cell must have to function and reproduce as a cell of a particular species. The E. coli has one circular chromosomes. That one chromosome is its genome. That one chromosome is approximately 4,000 base pairs long. Human genome consists of 22 different autosomal chromosomes and 2 different sex chromosomes. Moreover, each human cell, except the sperm and the egg cells, has a pair of each autosome and a pair of 6 chromosomes. The X, 2X, or 1Y plus 1X. Therefore, human cells are said to have a diploid genome. Bacteria have a haploid genome. A gene is a sequence of DNA which codes for a functional product. The product is usually a protein of some kind, like an enzyme or a receptor or a structural protein. A genotype. This refers to the genes which an organism actually possesses. This is a potential of the organisms. A phenotype refers to the genes which are actually expressed by the organisms. This is usually what the organism looks like. The allele. These are the different varieties of a gene which exists in a populations of the organism. So how DNA functions? Remember that DNA is a double-stranded, and the 2 strands are complementary. The strands are hybridized to each other because of the base pairing, and the 2 strands are anti-parallel. For each gene, one strand is positive and the other is negative. The positive strand is used to make the mRNA or the message ribonucleic acid. In a DNA replication, what is meant by leading strand and lagging strand? And where do RNA primer fit into this? The replication will need DNA polymerase, RNA polymerase, helicase, gyrase, and the ligase. The replication process ensures that when DNA is replicated, the 2 exact copies will be made, and if a mistake is made, it is called a mutation. A mutation is an inheritable change in the nucleotide base sequence. Many bacteria produces restrictions in the nucleus or the restriction enzymes, and these enzymes have become very important in tools in genetic engineering. And these enzymes open cut the double-stranded DNA at the palindrome sequence in such a way to generate the sticky ends. Such easily hybridized structures allow molecular genetics to connect DNA fragments together, splicing genes from different sources. And when the gene is activated, the mRNA is synthesized. This is called the transcription. The mRNA then is read and translated into protein by the ribosomes in a process called translation. In eukaryotes, there are often one more step between transcription and translation. It is called the RNA processing. Many eukaryotic genes are composed of multiple coding segments, which is the exons, separated by a non-coding segments, the introns. The complete gene containing exons and introns is transcribed. However, before translation, in protein synthesis, the transcription, the DNA is copied into mRNA or the tRNA. The RNA polymerase binds to the promoter and synthesizes mRNA from the positive strand of the DNA. Many copies of mRNA are made as the first RNA polymerase moves downstream around 1 byte. The RNA polymerase drops off when it hits the termination region. In protein synthesis, the transcription, the DNA is copied into mRNA or the tRNA. The RNA polymerase binds to the promoter and synthesizes mRNA from the positive strand of the DNA. Many copies of mRNA are made as the first RNA polymerase moves downstream around 1 byte. The RNA polymerase drops off when it hits the termination region. In translation, the mRNA is read and used as a template to make a protein. The 5' end of the mRNA binds to the small subunit of the ribosome, and then the large subunit binds to that. The AUG is always the first codon read. This codon touches the anticodon on the tRNA carrying methionine. Those methionine will always be the first amino acid of every new polypeptide. There are 61 different tRNAs. These differ in their anticodon sequence, which can hybridize with the 61 codons specifying amino acids. It follows then that the different anticodon sequences correspond to the different amino acid. The amino acid are attached to the 3' end of the tRNA strand. The mRNA binds to the ribosome so that the first AUG binds to the P site and the second codon is the A site. The two corresponding tRNAs come in peptidyl transferase, transfers, and covalently attaches the first amino acid to the second amino acid. The first tRNA leaves and the second codon moves to the P site and the third codon moves into the A site, and the process repeats itself.
So that's all for today.